The paper published today in Acta Neuropathological Communications entitled "The Dysfunction of the mTOR Pathway is a Risk Factor for Alzheimer's Disease" contains several layers of results and data. In the first instance, we have disentangled the molecular pathways that are regulated by mTOR. mTOR is a very interesting molecule. It is almost like a gatekeeper of messages that come from outside the cell, and it allows the cell to survive in the given environment that it has to live in. So it always modulates the responses of the cell to its outside environment. The molecular pathways downstream this molecule were not known until now. We managed to find these molecules that execute the message of the gatekeeper. The next level of analysis that we have performed was to look at how these molecules are deregulated in Alzheimer's disease. And what we found that in the brain of Alzheimer's disease patients, the molecules that execute the messages of mTOR are very heavily deregulated in the patients, and they are deregulated before the pathology develops in the brain. The third layer of data relates to the dysregulation of this pathway in peripheral blood cells. What we found a long time ago in 2001, that we can detect a dysregulation of this pathway functionally from blood cells. Now we repeated the experiment, and it works with a very high specificity and sensitivity to tell apart Alzheimer's disease patients from healthy elderly individuals. This means, taken together, that the dysfunction of this pathway, which we can detect from blood cells, is a constitutional characteristic of an individual, and it makes them very vulnerable to any kind of insult that affects their brain. And by insult, I mean aging, I mean elevated plasma homocysteine levels, lack of oxygen, lack of food, the effects of diabetes, etc., all the risk factors of Alzheimer's disease. And the individual may be born with this vulnerability, but it will not translate into a disease until these external risk factors affect the individual, thus the late onset of Alzheimer's disease. Collecting the data for layer 1 was pivotal for us in order to be able to carry out the research presented in layer 2 and layer 3 of the paper. But beyond that, that set of data carries an importance for the whole research community interested in these pathways. Until now, we knew the functional consequences of regulating this pathway because we had drugs that could interfere with mTOR. However, we never knew what molecules carry out these jobs that mTOR is doing for the cell. So the first layer of data kickstarted our own research, but it will be terribly important as a starting point for researchers looking at cancer, drug resistance to rapamycin in cancer patients, looking at mechanisms of traumatic brain injury, and a lot of other diseases where mTOR has been implicated by functional studies before. The layer 2 data is really important in order for us to understand what is the importance of this pathway for Alzheimer's disease. What we could disentangle here or understand with this study was that the deregulation of these molecules downstream of mTOR happens well before the pathology develops in the brain. So it's not a pure consequence of the accumulation of the plaques and tangles and nerve cell death that we see in Alzheimer's disease, but it is something that appears before the pathology develops. And all this has been made possible by the very large brain collection of the Thomas Willis Brain Bank in Oxford that collected brains from patients who have been longitudinally studied by the Oxford Project to Investigate Memory and Aging, the OPTIMA project. The third layer of data is data collected from blood samples from Alzheimer's disease patients and healthy elderly individuals who were age-matched for the study. What we have done is we repeated our study from 2001, looking at the functional integrity of the mTOR pathway from blood cells. Now, what we observed in 2001 was that the dysregulation of this pathway was strongly associated with Alzheimer's disease. What we managed to see now, that the dysregulation of this pathway indeed has a very strong association, indeed a more than 80% sensitivity and specificity for Alzheimer's disease patients relative to healthy elderly individuals. What this means is that we have a blood test that relies on measuring the functional integrity of the mTOR pathway, which can tell the vulnerability of that individual patient to any exposure to the risk factors for Alzheimer's disease. So for the individual patient, a positive phenoTOR test, as we call it, would mean that if they become exposed to oxygen deprivation to their brain, lack of nutrients in their brain, high homocysteine levels, or they hit their head, they have a head trauma, then the likelihood of them developing cognitive deficit and dementia is much, much higher than for a patient who has a negative phenoTOR test. So this blood test basically gives a measure of the vulnerability of the individual to these outside risk factors of Alzheimer's disease. We have to do a lot of things. When you find a vulnerability factor or risk factor like this, what is the next important question to answer is how big a risk this factor represents. Does it mean a 5-fold increase in risk, or does it mean a 50-fold increase in risk? To be able to determine the strength of the risk, we have to have a lot more patients. So the next step for us will be to carry out a very similar study to layer 3 of this paper with a large number of Alzheimer's patients and healthy elderly individuals. Once we determine how big a risk this dysfunction represents in Alzheimer's patients relative to controls, what we have to do is a longitudinal prospective study looking at patients with mild cognitive impairment, because this is the patient population that presents with very mild symptoms. The diagnosis is very uncertain. You don't know for sure what's going to happen to them next. So using a vulnerability detection or a risk detection test could make a real difference for the clinical management of this patient population. But in order to provide clinical qualification for a test, you have to carry out the longitudinal prospective study on a large patient cohort, looking at how this risk factor relates to how fast cognitive deficits develop, how severe the cognitive deficit that develops is in the patients with the positive Phenotor test. So that will be 2 very large clinical trials that need to be carried out in the near future.